Hey class, we're back here in the lab today and we are going to be going over how to make dilutions. This is usually in an exercise in your book all together with dilutions, NPNs, and pour plates, and it is about bacterial enumeration. And so what that means is that we are trying to get a count of how many living bacterial cells are in a sample. For an NPN, we are looking usually mainly at E. coli numbers. So E. coli is a fecal coliform indicator, and what that means is that if E. coli is in a water source, then that tells us that that water source has been contaminated with feces, because of course that's E. coli's natural habitat is the intestinal tract of animals, including us. So getting a count of how much E. coli is in a water source is important because that can tell us if a sewer system is working properly or if there's some contamination that's going on. Pretty much every river system or water source is going to have some number of E. coli in it, but we don't want those levels to get really high. So we have to be able to get a count of how many bacteria are there. But before we go over the NPN and the pour plate technique, which I will do a separate video for, I first wanted to do a video on serial dilution. So how do we make a dilution? And the reason why we have to do dilutions is because bacterial cell numbers are so high. Okay? In a 100 ml sample, which is not a whole lot, you can have millions, tens of millions, hundreds of millions of bacterial cells in that small sample. So we can't accurately get a count on how many bacteria are there with that high number. So what we have to do is we first have to dilute the bacteria. Then we can do the test on it and then take into account how much we diluted it. That's called our dilution factor, to get an estimate of how many bacterial cells were in the original sample. There is a diagram in your lab manual very similar to what I have up on the board. So I'm going to go over that, and then I'm actually going to do the dilution for you, showing you exactly how you would be doing it if you were in the lab with me. So starting out here, we have so there's a few things that you need whenever you're doing a dilution. Number one, you need your sample, your original sample. So I have an original E. coli sample that we have in the lab here that we've inoculated. So here's my E. coli sample, and this is a 10 ml sample. Okay, so it's 10 mLs. So we need our original sample. Now normally if you were doing a count, you would be getting it from a sample. Okay, from the environment. But here we're going to use our lab E. coli that we have. So you need your original E. coli sample, and then you also need what are called dilution blanks. So these are blanks that are sterile, so that means there's nothing in them, and then there is a buffer in them. Okay, so these are bought dilution blanks, so they're already prepared, and there's 99 mLs. That's going to be important, so you need to remember that. There's 99 mLs in one dilution blank. Now, we also can make them in the lab, and they're put in what are called prescription bottles. So it's just 100 ml bottles. And then we put the same buffer that's in here, and then we autoclave them to make sure that they're sterile and that there's nothing in them. So we need dilution blanks. We need our original sample. We're also going to need a pipetter. So I have an automatic pipetter here that I'm using. If we were doing this in our actual lab, you would be using a manual pipetter, and that's where you have to roll it up, and it's a little bit more difficult to use, but since I'm going to be the one that's doing it, I use my nice fancy automatic pipetter. So whenever you press the top button, it sucks up. When you press the bottom button, it pushes the liquid out. And then you need your pipettes, okay? So you're going to be sucking up liquid, so we're going to be using our pipettes. Now there's different size pipettes. This is a 1 ml pipette because that's how much I'm sucking up. It's going to be 1 ml. There are 5 ml, 10 ml, 100 ml, different size depending on what you're doing with them, but we're going to be using a 100 ml pipette. And there's two parts to the pipette. There's the top part that has cotton at the top. So that helps prevent contamination because we don't want to suck up E. coli into our pipetter. And then there's the bottom, and that's where the liquid comes out. So if I fill this pipette all the way up to 0, that is a full 1 ml, okay? So, and that's with any pipette. So you fill it all the way up to 0, that's going to be a full 5, 10, 100, 25, whatever you're pipetting, okay? And it's broken up into tenths. So if I were to suck up only to the 0.9, that's only 0.1 mLs because this is a 1 ml pipette. So I've got my pipetter, pipettes, my dilution blanks, and my E. coli. So now let's talk about what we're going to be doing. So here's my original sample. Here's my dilution blanks that I have drawn on the board. Don't judge my drawing. I'm not an artist. Okay. So the first step is going to be pipetting from my original sample. So I'm going to pipette 1 ml into my 99 ml blank. So 1 into 99, so 1 over 100, because I pipetted 1 into my 99, so now I have 100 in there. 1 over 100 is 10 to the negative 2. So what that means is that from my original sample into this dilution blank, my sample has now been diluted 100 times. Because I put 1 ml into 99, now I have a total of 100 mLs in there. So it's 1 over 100, which is 10 to the negative 2. Now, remember I told you there could be tens of millions of bacterial cells in this original sample. So simply diluting it 100 times is not going to be enough. That's not going to dilute it enough to where it becomes workable numbers. So we need to keep going. So from my 10 to the negative 2 dilution, I'm going to give it a little shake, and then I'm going to take 1 ml out of my 10 to the negative 2 and put into a new dilution blank. So I've taken 10 to the negative 2, 1 over 100, and put it into another 99 ml blank, 1 over 100, which is 10 to the negative 4. You multiply these. So now, my original sample, if I move all the way over here, has been diluted 1,000 times. 1 over 1,000, or 10 to the negative 4. Again, most of the time, depending on which sample it is, that's not going to be enough. I'm going to need to keep going. So I do the same thing again. Give my 10 to the negative 4 dilution a little shake. Take 1 ml out of it, put it into a new dilution blank. You've taken 10 to the negative 4 and now put it into a new dilution blank. So now your original sample has been diluted 10 to the negative 6, which is 1,000,000 times. Okay, so this is where we're getting to now. We can get to use workable numbers. So every time you move, you take 1 ml out of the previous sample, you're diluting another 100 times, 100 times, 100 times. Okay? So now I'm going to demonstrate how we do this. All right, so here are my dilution blanks. I've already got them labeled. So I've got 10 to the negative 2 all the way to 10 to the negative 10. When we're doing our NPNs and pour plates, which we'll talk about in a minute, we want to dilute to extinction. And what that means is we want to dilute all the way till we have no more bacteria left. And that's why I'm going all the way down to 10 to the negative 10. I've got my original E. coli sample, my pipettes, and my automatic pipetter. So what I always like to do is have these lined up and ready to go. Okay, these are bought, so I'm going to have to open them. I'm going to go ahead and do all of that. That way they're all open. And then we'll start pipetting. You're not using the Bunsen burner for this or a loop or anything like that. You're using pipettes, and they are disposable. So once I'm finished with them, I'm going to put them in the biohazard. And you want to change out your pipettes with every dilution. You don't want to be mixing your dilutions. All right, so first, here's my E. coli. I'm going to give it a little bit of a stir because we want it to be evenly distributed. We don't want to have a whole bunch on the bottom and not a lot at the top. All right, so I've got my E. coli nice and mixed. So the way that I like to open these pipettes is just tear the paper, making sure that I have the cotton tip, okay? And you don't want to touch this part, okay? Remember, aseptic technique. You do not want to touch the pipette itself. If you do that, then it's not sterile anymore. So you're going to insert the cotton part into the automatic pipetter, and you're creating a vacuum, so you want to push it kind of hard. What I always like to do is I always keep the paper on the pipette until I'm ready to actually pipette over. This helps prevent contamination, because if I want to lay this down to open up my dilution blank, I can do that. So I'll leave my paper on. I'm going to open my dilution blank, and I'm going to go ahead and pipette 1 ml into my dilution blank. I always put it back in the paper so I'm not dripping E. coli everywhere, and that's going to go in the biohazard. Now, I close my 10 to the negative 2. Really good. And I'm going to give it a good shake. So now, going from this, which is my original sample, to this, the liquid that's in here, the cells that are in here that have been diluted 100 times. Okay? So this is a perfectly clear one. This one has the cells in it. So you can tell that it's kind of cloudy and that there are some cells there. So now I'm just going to keep doing that. I'm going to go take 1 ml from my 10 to the negative 2 and put into my 10 to the negative 4. So I'm going to use a different pipette. I'm going to give this another little shake. Open my 10 to the negative 4. 1 ml. So now I'm finished with my 10 to the negative 2. I don't need it anymore. I'm going to open my 10 to the negative 6. Give my 10 to the negative 4 a little shake. Mix those cells up. Now I'm finished with the negative 4. Same thing all the way down the line. And the last one is my 10 to the negative 10. And by this time, hopefully I've gotten to extinction where there are no more cells left. Okay, now I am finished. So I have all of my dilutions here. Right now you can't really tell the difference in them unless they're labeled, so that's why it's important that you always want to label all these. So I've got my 10 to the negative 2 all the way down to my 10 to the negative 10. So you would do this to make your dilutions, and then now once we have our dilutions made, we can move on to doing our MPN and our pour plates. We'll read those results, and then we'll be able to factor in the dilution factor and then figure out how many cells were in this original sample. Okay, so I will post a separate video for the MPNs and the pour plates after this one, after the dilution.